Okay. So the green sulfur and green non-sulfur bacteria, though, except for particular kinds of environments, are a minor player in the microbial world, minor players. We've reached now the first group of organisms, the first bacterial phylum, that is a really major component of the biosphere, the cyanobacteria, blue-green algae. And the general rule is if there's water, there's cyanobacteria. There are a few environments, up above about 80 degrees, there aren't any. But there's cyanobacteria that live in Antarctica. Cryptoendolithic cyanos. They live down inside the surface of the rock, collecting light through the crystals. Yeah?
I mean, we have cyanobacteria.
You have them in your hair.
Not like in our mouth.
Only probably fortuitously, pollen and that kind of stuff. There's some symbionts in this group, lots and lots of symbionts. I'm not aware of any pathogens. There are some organisms that are pretty awful, though, that produce some pretty bad psychoactive and neurotoxins in this group. There are thousands of species in this group, and so I only provide you a few of them. These things are, on one hand, they're phenotypically homogeneous. All of these things are photosynthetic. They carry out the Calvin cycle. They have two photosystems, photosystem 1 and photosystem 2. Most of them have bacterial chlorophylls. There's one group called the prochlorophytes that have, I hate to call them bona fide chlorophylls, but the traditional eukaryotic type chlorophylls. And so metabolically, they're all pretty much alike. Most of them can fix, many of them can fix nitrogen. They're all photoautotrophs. Morphologically, they come in every shape and size you can imagine and live in a wide range of environments. Most of them, though, although there are thousands of species of these, most of them don't have species names. Most of them are referred to by their genus name and a PCC number, the Pasteur Culture Collection. And so you see things like Anabina PCC 6309. That's the common way that the cyanobacterial species are referred to. This is actually a plant culture collection, and historically, all of that included the cyanobacteria as well. They have two photosystems, so they carry out Z-scheme oxygenic photosynthesis. So they make oxygen. It's an interesting conundrum that these organisms have. They can fix nitrogen, mostly. The enzyme that does that is called nitrogenase. Nitrogenase is extremely sensitive to oxygen. I'm sure you can imagine why. This is nitrogen. This is oxygen. They look a lot alike, right? And so an enzyme that binds to and reacts with this is also likely to bind to and react to that. This is a futile cycle. This is photorespiration, right? You've heard about that in plants, and it's a big waste of energy because this fixing process takes a huge load of reducing power to function. And if you're doing that with oxygen instead of nitrogen, you're wasting all that energy. And so plants have these complicated ways to keep this nitrogen fixation process away from the oxygen generation process that's photosynthesis. And these organisms have the same problem. How can you fix nitrogen, but also be making lots and lots of a potent inhibitor of that nitrogen fixation? And so there are different ways that these organisms separate these processes. One of them is to separate the processes in time. You can grow photosynthetically during the day and then fix nitrogen at night. There's a bit of a problem with this, which is that nitrogen fixation takes so much energy that you're likely to run out of energy before night falls, night's done. And so you'll fix nitrogen for a few hours, and then you're out of energy, and you have to wait for the sun to come out again to get more energy. Some organisms separate them physically, and so they'll be in a filament, and you'll have most of the cells will be photosynthetic, but occasionally you'll have a cell that's specialized for nitrogen fixation and not photosynthesis. They do carry out phototrophy. These things are called heterocysts. They carry out cyclic photophosphorylation, but they don't carry out oxygenic phosphorylation. And so they don't generate oxygen, and they keep the nitrogenases functional there. You guys have probably heard all about this in bacterial physiology, microbial physiology, etc. It's a really interesting process. It is a terminal differentiation. Those heterocysts, once they start the cascade for differentiation to a heterocyst, they can never go back. And they can never divide. It's a terminal differentiation. That's the definition of a multicellular creature, where you've got some cells that will go on to the next generation. These are germline cells. And some cells that cannot go to the next generation. So a heterocyst-containing filament of Cyanobacterium is a multicellular object. It is not kind of the traditional unicellular thing that you think about. It's really one good example of that. And you guys know all about how they trade resources around in the periplasm. It's pretty cool. These organisms typically have thylakoids. This is where thylakoids come from. And they're flattened against the cytoplasmic membrane. They come in the families are different based on how they reproduce. One group, these are unicellular, they typically are embedded in capsules. They divide by fission or by budding in irregular groups. This is microcystis, and most of you have seen microcystis before. If you go to a eutrophic lake or river, you oftentimes see this brilliant green sheen on the upper surface of the water. A lot of times you see this around places where birds are. I know there's some good places in Greensboro to see them. I don't know where they are around here. But it looks a little bit like green paint on the water. That's microcystis. Microcystis produces a neurotoxin, and occasionally a deer or something will be intoxicated by this. They drink the water, they suck up a bunch of microcystis, and you end up with a deer with liver failure. Occasionally a person will get microcystin intoxication, but it doesn't happen very often. The plurocapsule ones like Dermocarpa, these organisms divide by multiple fission. And so Dermocarpa is a great example of that. You've got an individual cell and it gets bigger and bigger and bigger and bigger. And then it divides into lots of cells all at once. These smaller cells contain multiple nucleoids, but they flow freely in that cytoplasm. So they're syncytial cells. Every cell has several nuclei. And then they divide all at once like this. Many of you have seen oscillatoria, maybe in your winographic column. If you don't, you probably will. These are filamentous organisms made up of individual cells, and a lot of times they are really thin cells, so they stack up here. This is like a big stack of quarters, right, of individual cells. No differentiation, one cell type to the next, except for the morphology of the terminal cell can be different. These things have, this one has a sheath. Sheaths are common. They're called oscillatoria because of the way they move. These filaments are motile by gliding. And that gliding motility causes the filament to rotate as it moves across the surface or through the liquid. And if the end has any irregularities in it, which they usually do, then then they seem to wave back and forth as they glide through the solution. They're not always thin cells like this. Sometimes they can be longer rod-shaped cells or even almost cockeyed as they in these chains. The nostocs look a lot like that, but they have differentiated cell types. And so this one is Anabina. This is the one we just talked about a little while ago. Here's a heterocyst. Notice the difference in color. This is another heterocyst. When you see Anabina filaments, very often, oh, and these are baecytes or aconites. So it's basically a resting phase. Very often these filaments end in either an aconite or a heterocyst because the connection between cells is weakest there. Aconites are formed during starvation. Heterocysts are formed during nitrogen starvation. There's also a hormogonia, which is a, what's the other one? Aconites, resting stages. There's another cell type that these things have. They are hormogonia. And these things are chemotactic. A lot of these organisms are symbionts of plants. And so you see them on aquatic plants as a green film at the water line. The most morphologically complex are Fissurella and its relatives, all this, I can't even pronounce that, going on the tail leaves. All the organisms we talked about so far that are filamentous are linear filaments. And they're that way because they divide, the division plane is always perpendicular to the axis of the filament. These organisms also have divisions that are parallel to that filament. And so you end up with filaments made up of several cell diameters. And then often one of the cells in that filament will divide and form specialized cells that form these branches. So organisms in this group form branched filaments, complex filaments. And in fact, they're usually mistaken for eukaryotic algae, which they are not. These things are modal by gliding. Believe it or not, this whole thing can glide in one direction or another. They also have heterocysts. They also have aconites, and they're usually sheathed. There's usually a sheath that surrounds this main filament. The last group we're going to talk about are the proporococales, or proporals. These organisms they're typically morphologically simple. I mean, proporon is just a coccus, right? The distinguishing feature here is that these things, all the other organisms have bacteriochlorophyls. These ones have chlorophylls. And these are related to chloroplasts. And so chloroplasts are in this group. And many of these are symbionts of other creatures. So organisms in this group that form symbiosis with corals. Proporon itself is a symbiont of ascidians, sea squirts. And you see them on the reef, these nice little green, grape-looking things. They are external symbionts, they're not endosymbionts. And they live typically in the inside body cavity of the animal. These organisms are essential for the health of the animals. And so in the case of proporon, if the proporon leaves, the ascidians die. They just can't get enough energy by filtration. Coral reefs are pretty pristine environments, right? There isn't that much organic material that the squirt can filter out. And so it has to supplement that by photosynthesis or it can't survive. Members of this group are quite abundant in the sea.